Okay, so a second here, I'll share my screen. Okay, so today we're going to be talking about these topics: sampling, filters, convolution, and digital formats, what the data looks like when it's on the inside of a computer. Okay, so last time we talked a lot about signals, voltages that vary as a function of time. So today we're going to talk about Fourier transforms and so forth, which is something I'm sure that all of you probably heard of. Maybe you're not. Some are, I'm sure, much more familiar with than others. The idea is that almost any signal, and I should say any signal that you'll encounter in real life, can be decomposed into a sum of sinusoids at different frequencies. And so here's an example where you can see this square wave, and you can see that if you have just one sinusoid, the red one, you get a certain approximation, degree of accuracy, and then the yellow one, you add a second one, and the third one. The more sinusoids you add, you eventually, it gets closer and closer approximation until you have an infinite number of sinusoids, and it's perfect. And so this is actually a nice example of that I found. And so if you look at a circle, and we'll talk about this later if we have time, if you have a circle and you just have a point, you have a point going around the circle, then if you look at the projection, so say this red sinusoid right there, if you look at just the vertical position of the point right there that I'm following on the circle, that is that sinusoid. So sinusoids are just when you trace a circle around, and we'll talk about that. So each of those circles is a sinusoid of a different frequency that's tracing around and then drawing out this square wave. All right. So the idea behind that is, so this is everybody is sort of familiar with this with audio, that you have bass and you have treble and you have mid-range. Right, so the bass would be the very low frequencies that vary slowly, and treble, of course, is the high ones. So the Fourier transform, which we might or might not talk about later, depending how much, in depth, depending how much time we have, converts from what's called the time domain to the frequency domain. So the time domain is what we're all familiar with: the voltage as a function of time, or some signal as a function of time. And then the Fourier transform is its representation of frequency space. So here we have time on this axis, and then we have intensity, however you want to phrase it, on this axis. Here you have frequency on this axis, and then how much of the frequency is up here, right? So this is frequency of 0. So one thing that's a little bit maybe counterintuitive about this is that you have positive and negative frequencies here. So you could just look at half of it, but the reason you have positive and negative frequencies is that these values here are actually complex values. So if you have a real valued function, so pretty much all of our signals are real, real as in no imaginary numbers involved. So complex number, for those of you who might not remember, it's when you have a real number plus an imaginary number. So a plus bi, I being the square root of -1. So the coefficients for each frequency here are actually complex numbers, or even if this is real. Okay, so the reason we have these positive and negative frequencies, way to think about it, it's a way for the, if you do it like this, where one is up and one is down, or rather the coefficients are complex conjugates of each other, then those cancel out, and then you get a real valued function. But in any case, that's the idea here, is positive and negative frequencies, which are the same. So if you give this pulse right here, so this is sometimes called an impulse function, or a Dirac delta function after Paul Dirac. It's just something that's sort of, it's a function that's infinitely narrow and infinitely tall and has an integral of 1. And so it's just a blip. And so if you want to characterize the system, you can sometimes just give it a blip and see how does it respond to a blip. And a way to think about that is something like action potential. So how does GCamp respond to a single action potential? Anyway, so the frequency domain, one thing that's relevant here is that the delta function contains all frequencies. And one of the reasons that's relevant is that if you have a linear system, we'll talk about in a minute, and you know what its impulse response function is, meaning its response to a single blip like that, then you can predict its response to any function. So if you know what GCamp, the response to GCamp is to one action potential, then you could predict the response of GCamp to any train of action potential in theory, right? So that's not including things like saturation effects and so forth. Okay, so anyway, so that's maybe all you have to know, that there's a time and frequency domain. All right, is this clear so far to people? Any questions? We'll talk about it more in depth and maybe even have equations later if we have time. All right, so filters and transfer functions. So the way to think about filters mostly is that they're things that block certain frequencies and let other frequencies go through. So I couldn't find really a good demonstration of that, so I'm going to try to draw it on the screen. So by that I mean, imagine you have, let's make this relatively concrete. Okay, so imagine that you have a recording from hippocampus, something, right? And in hippocampus you have the theta oscillation at 8 hertz, right? And then you have action potentials that occur on top of that. Okay, so your signal looks something like that. I'm actually going to see if I can make this smaller. Okay, there we go. So all right, so we have action potentials on top of that. Right, so if we were to look at in the frequency domain, where here is the frequencies, and then this is the amount of, I don't want to get, you can think of it as well, we can call it power. We can talk about that later. But anyway, so this is 0. So the amount of power, or the amplitude of the coefficient there, the magnitude of the coefficient is how, essentially a reflection of what the mean of the signal is. So we're going to have some sort of, so if this is 8 hertz, okay, we're going to have some sort of thing that goes like, you know, and then it's going to have some big bump at 8 hertz. And then it's going to kind of fall down, and you have this high frequency content out there. All right, so this is not a linear scale. So let's say that's like 300 hertz. Okay, so so a transfer function is something that if you take that in that representation in the frequency domain, and then you can multiply it by something called, for example, a low pass filter. And a low pass filter is something that looks like that. So that would be its transfer function, by which I mean that if you multiply this times that, then what you'll get is something that looks like, you know, like that, right? And so out here is the spikes, and then here is the theta and other LFP features. So we'll talk a little bit later. I mean, when we actually talk about electrophysiology, they don't separate nice and so cleanly, but for theta they do. So in this case, if we were, so this is a low pass filter, meaning that the low frequencies pass, and then this is called the pass band and the stop band, right? So when you have a low pass filter here, so what if you were to look at what that signal would look like when it's low pass filter, basically these spikes would be removed. And whereas if you were to do a high pass filter, then it would be something that sort of looks like this. And then if you multiply this times that, then you get something that looks, you know, sort of like that, and then goes up, and then you have that stuff. Okay, so which is your spikes, right? This is obviously a bit of an oversimplification, but then the high pass filtered version looks something like this. And so typically when people do, we'll talk more about electrophysiological recordings, but this is one of the things that people do. But anyway, conceptually, so does everyone get the notion of, say, high pass and low pass filtering, the notion of the transfer function? Yeah, okay. All right, so to here. Now, and I actually have an example. So this is some sort of earthquake data. That's, I guess that's the, you know, the displacement of some probe or whatever, and then you see that that's what it looks like over time. And here you can see the low pass filtered version. It's harder to find examples of high pass filter things. That's why I drew that. But something that a clear example of something high pass filter would be a train of action potentials where you're trying to, for example, spike sort them out from, you know, out from without the big LFP fluctuation. Okay, so well, here's another good example. So you can do spatial filtering. So it doesn't have to be time and frequency domain. It could be space and spatial frequency. So this is an example of if you low pass filter so the standard deviation here is a standard deviation of the Gaussian in pixel. So they do this filtering. They actually do this. Well, let's talk about that later. But anyway, so this is sort of low pass filtering where you're keeping some of the middle frequencies, and this is ones where you're keeping really just the lowest frequencies.
Hey, Luke. Yeah. I have an example of high pass filter, like I'm recording right now.
Okay.
I can show it. I don't know.
I actually have one I can show later.
Oh, okay.
So Maybe. So when we have the lecture actually on data analysis for the ePhys data, which I have not planned at all, we'll have to figure out some way to have people screen share and show stuff like that. So let me talk to you later on for next week.
Sounds good.
All right. All right. So now we're going to talk about. Sorry. Go ahead.
What is the standard deviation exactly?
I'll get to that in a second.
Okay.
So okay. So you can do filtering in the frequency domain. You can conceptualize filtering in the frequency domain the way that I just showed it, but you can also do filtering in the time domain. And so the key idea here, is this notion of this operation called convolution. Okay, so f here is supposed to be the function, f is a function of time. So this was originally for space, and I just changed it to time. But that's, so like I was saying, that with an image, it's typically space in terms of like number of pixels across. For, like physiology, for example, is time. But the idea here is that f and g are both functions of time, whereas big F and big G are, this is the Fourier transform of f, so this would be f as a function of frequency. So if you multiply f times g, so say that this is the function and this is your filter transfer function, that's actually equivalent in the time domain to convolving f with g. And that's the convolution operator, is this x with the circle around it. And it's actually the same thing in reverse. So what that means is that we could either, say Fourier transform stuff and, multiply these things together and transform it back to filter something in principle, or we could convolve f with essentially the Fourier transform of the filter's transfer function, which is something that's often called the convolution kernel, or the filter's kernel. Okay, so I'm not going to write down the equations and stuff because I think it's not even necessary, but I think that just to give you a conceptual idea of what convolution's like, this is kind of messy, I just wrote this out. All right, so linear time invariant systems. Remember we talked about, I can't remember if we talked about that last time. A linear system is one. Did we talk about linear systems last time? No? Okay. So it was in the reading though. That's what, okay. So yes, it was in the reading. So a linear system is one where the big thing with linear systems is that if you multiply the input times 5, then you get the output times 5. And then the second thing with linear systems, the key principle is that if you pass two inputs in, and you get, say if this is the input and this is the output to that input, and that's that input, and if you pass the inputs in together, you just get the sum of the outputs. So what that means is that when you add multiple signals together and pass them through, they don't interact with each other in any way. So what that means is that any, oops, this one, okay. What that means is that any input can be broken down into parts, for example, such as sinusoids. And then you can essentially, if you were to pass each sinusoid through one at a time, then you would know what the response to the whole input would be. And so that's what I was saying. If we know what the impulse response function is, we know what that output will be, which should be for any input, because you can imagine this broken up just down into those impulses that are shifted in time and scaled in size. Okay, so to get a little more, so anyway, so that's the notion of convolution. The best way to think about it is that if you have an input, and then you have this thing, which is the impulse response function, then your output is basically, so convolution basically takes every time you have a bump like that and replaces it with that thing. Okay? And so if you have a bigger bump, then replace it with the higher, if it's twice as high, then you get this thing, 2 of these. And if you have 2 bumps like that, then you get something that's the sum of both of those. Okay, so any instrument that you have will have will not have a perfect response. So for example, if you take a microphone and you have some sort of perfect click, microphone's response is still going to look something like that, where this is going to be, extremely fast, microseconds or something, but it's still never going to be exactly the same. So other examples of this that are more concrete will be, the clearest one would be something like GCAM, right? So if you have an action potential, then the actual calcium signal you get looks like this. You don't get a calcium signal that looks like that. Right, and that's for 2 reasons. One is that the calcium signal itself to an action potential would look something like this, and then there's the kinetics of GCAMs binding to the calcium and then unbinding of the calcium. So and we'll talk more about that in depth in lecture 5. But the idea here is that you can perform an operation called deconvolution, where you can attempt to take this output and then make an estimate of what its input was. So for GCamp, that would be to try to estimate as best you can what the train of action potentials was that generated the calcium signal that you saw. Another example of this is that in microscopy, there's something called the point spread function of a microscope. So if you have a tiny bead that's smaller than the resolution of the microscope, and you image that, you don't actually see a tiny point. You actually see this little blurry cone type thing. That's the impulse response function of the microscope. So it's sort of like if you image that, you see something like this. It doesn't look like that, but you see something that's blurred out like that. And then the idea is that you can actually perform deconvolution with your microscopy image to try to estimate the original distribution of fluorescent molecules in the sample, which is a little bit different than what the microscope actually shows you. Okay, so let's see what I was saying. Okay, so the idea here, so convolution is something we do in the time domain, and it applies to lots of things that we do in neuroscience, such as GCamp signals or channel rhodopsin signals or point spread functions of microscopes, things like that. But filtering, as I said, also can be done by convolution, right? So this is the transfer function of a low-pass filter, and that's what this is. So if you Fourier transform, then you basically get this, which is the convolution kernel. So if you convolve some function of time with a Gaussian, so say this is approximately Gaussian. So with a Gaussian like that, then you get it blurred out. So to answer Elliot's question from before, what was the standard deviation in the image of the hot air balloon? It was the standard deviation in pixels of the Gaussian that of the two-dimensional Gaussian that they convolve that image with. So, so does that make sense for people? Be honest if it doesn't, please.
No. Can you draw it like on the graph?
Yeah, sure. Yeah. Right. So the idea is that if you were to, or imagine if you had some sort of thing like this where you have, where you have a train of action potentials, right? Those could be, say, action potentials or pulses of light, right? And then you have, if the impulse response function of GCamp, for example, is something that looks like that. Maybe actually let's make it more like that, longer. So then what you'd have your calcium signal would be something that looks like this. It'd be something that looks like, see where's the sum of those different things. So it's essentially if you take this right there, that's what that is, and then you just have more and then they add together. It's not a very good drawing, but I think you get the point. So what I'm saying is that you're convolving with this kernel that's actually equivalent to filtering this signal, and the filter that you're applying is the Fourier transform of your convolution kernel. So what I'm saying is that if you take the on and off kinetics of GCamp, right, and it looks some sort of, and it's this double exponential type function where it's exponential going up and one coming down, then if you were to take the Fourier transform of this, then that would actually be the transfer function of the filter that you're using. So GCamp actually filters the spike train. So the calcium signal is a filtered output, and it turns out that this type of thing is pretty much that its transfer function is pretty much a low pass filter. So what GCamp does to a spike train, the GCamp calcium signal is actually a low pass filtered version of the spike train. Because in the time domain, it's this convolution, but that's actually equivalent to filtering in the frequency domain. Okay, so so there are different pros and cons to this. So I guess one thing that is, all right, so let me talk about the image. So the image, imagine, you have sort of image like this and has a bunch of pixels, right? And then there's each pixel has a value, something like that, right? So if you imagine if we just, so it's two-dimensional, imagine you just take a slice through the image like that, and you look at it, and then you have some sort of thing that looks like, I don't know, whatever, something like that. So, and then you convolve that with a Gaussian filter where the standard deviation of that thing, as they say, was like 3 pixels. And so then what happens is you'll get some sort of thing where so you get some sort of blurred thing like that. If you look from the side, maybe it would actually be maybe blurred more than that. But then if you look at that, that's what And a two-dimensional version of that is this. We have the standard deviation of 3 pixels or standard deviation of 10 pixels. So that is, so what you're essentially doing there is you're low pass filtering, except you're doing it in the convolving with a Gaussian distribution, you're just doing it at the time domain. Okay, so and those are what filter kernels will look like for these different high pass and band pass filters. So band pass lets one frequency through, low pass sort of only lets low ones through. So in a perfect world, I should say that a perfect right, so perfect low pass filter would look something like this, right? And then that's some sort of frequency that's a cutoff. And a perfect high pass filter would look like that. A perfect band pass filter would just be like that or something, right? But in practice, filters are so like sort of really crappy filters are like that, and then you can sort of higher order filters look more like that. But it's never a perfect thing. Anyway, so yeah, so one relevant aspect of this is that this may be a little bit too much detail, but convolution is computationally very intensive, or at least it can be. So a lot of times it can be computationally much cheaper to perform a convolution in the frequency domain. And the reason why is because the fast Fourier transform algorithm was this groundbreaking algorithm that allows you to do these processing like order n log n and sort of n instead of n squared steps. And so anyway, sometimes it's actually faster to Fourier transform and then multiply and then go back than it is to do the convolution in the time domain. So if you use like the conv function for convolution in MATLAB, I'm pretty sure it does in the frequency domain and then transforms it back. Anyway, okay, so does everybody get this sort of duality, convolution and filtering? Okay, okay, harmonics and notch filters. Okay, so this is relevant in electrophysiology, and we'll talk more, maybe not just even in electrophysiology, but I don't understand why that happens. Anyway, whatever. Somehow it gets drawn and it doesn't go away. All right, so all right, so if you imagine you have a signal that, okay, if you have a signal that's a perfect sine wave, let's say that's at 60 hertz, then what its Fourier transform is going to look like is going to be just something like that. And of course, that number is actually a complex number, but let's forget about that. Okay, so it's just totally flat, and then this one narrow spike at 60, and that's it. Okay, so 60 hertz I mentioned because 60 hertz is the in the US anyway, that's the current in the walls is a 60 hertz. So you commonly get 60 hertz noise in electrical recordings. And in the EFIS lecture, we're going to talk all about that and grounding and getting rid of 60 hertz noise. And it's kind of 50% science and 50% voodoo. But one of the things in practice that happens is that a lot of times if you get a distorted 60 hertz wave where it's sort of, you know, not perfectly sinusoidal. I mean, maybe the simplest example would be, imagine you just had a square wave at 60 hertz. Then what actually you get is you get harmonics, where you get at 120 hertz and 240 and so forth, at multiples of 60. You get you also get this content. So you get these. So this is often called a spectral line, meaning it's a line in the power spectrum. So the power spectrum, I should say, is, well, the power spectrum is the real valued function. It's basically the Fourier transform squared. So it tells you just how much of each frequency is present, but it doesn't tell you the phase information. The phase information is stored in essentially the complex part of it. But anyway, the idea here is that you have these, and these are called harmonics. So what often happens with 60 hertz noise is that you get something that looks, you know, kind of maybe like more triangular or something like that, and that will have power out here. Okay, so as a notch filter, a notch filter is a filter where its transfer function is like this. It has a notch. And so an example of a notch filter would be something when you want to filter out 60 hertz noise. So you can, if that's your Fourier transform, and then that's your thing, then basically you should get, you know, you should have your signal and it should, you know, be like that. And there should, and actually there'll be a little bump like that at 60 hertz. So notch filtering, and then you might have to notch filter also with the harmonics. Okay, so notch filtering can come in handy. What I'll say about it is that, so it's useful in certain situations and not others, and the situation in which it's not, a situation in which it's useful is if, say, you're recording something like action potentials, where virtually everything that you need to know about the action potential waveform is above 300 hertz. It's in a range between, say, 300 hertz and 8 kilohertz, right? So if you, so actually it is possible you could have harmonics that get up into that range, but if you filter out 60 hertz, and you know, so say the original signal looks like this, that's at 60 hertz, and you have spikes on top of it, right? Then you can notch filter at 60 hertz, and you'll get your perfect action potentials, and that's fine. The problem comes when, so imagine that you have, that you actually want to record the local field potential and the, you know, and the spikes, right? So imagine that you have, you know, hippocampus where you have, you know, where you have gamma on top of theta, and then you have spikes. So you have the theta 8 hertz rhythm, which is at 8 hertz, and then you have gamma up here, which is around, say, like, you know, I don't know, say 40 to 60. Something like that. So your actual power spectrum there is going to be something that looks like this, and then you have this big peak around 8, and then you have this more sort of peak around there, and then you have all this stuff, which is your spikes. And high gamma and spikes put together. What I'm saying is that if you have 60 hertz noise, then all of a sudden, boom, you have that thing right there. And then if you notch filter that, then you're going to get something that looks like this, and right. And so you've actually thrown out your signal, the actual physiological gamma signal, you know, gamma oscillation, which is in the same frequency range as the 60 hertz noise. Then you've gotten rid of that. So that's why this sort of filtering can be bad. So there are methods that you can use, computational methods that you can use post hoc to remove 60 hertz noise that do a much better job than a hardware notch filter. I won't go into those, but there are ways that, so that was actually, I will say briefly that it's interesting why they were developed. And the reason they were developed, it was this guy Thompson developed this thing called multi-taper power spectral estimation. And the reason was because he was a geologist or something, or earth scientist. And they wanted to study, they wanted to see if global warming was real. This is like in the 80s. And the problem is that if you actually put just thermometers on the surface of the earth, what you get is some sort of thing that looks like this. I'm not doing a very good job, but you have these oscillations on the period of 24 hours, and then you have these other oscillations on the period of 365 days. And then I think you also have oscillations on the order of like 10 years, or, you know, cycles and stuff like that. So if you actually look at if you were to plot the power spectrum of Earth's temperature, right, that you get some sort of thing where you have this you know, we have this very low frequency annual oscillation, and then like diurnal oscillations, maybe other oscillations or whatever inside. And just average, it's actually, if you just average the temperature for a year and average it for a different year, the amount of variation contained in these oscillations is bigger than the amount of temperature change from year to year. So it actually makes it so you can't just average temperature over a year and see if global warming is occurring or not. And so, so they actually came up with this method to remove spectral lines so that you could then demonstrate that global warming was occurring. So anyway, that's, you don't need to get into all that, but I just think it's interesting. But the point is that the best thing to do is get rid of 60 hertz noise by optimizing your recording setup. Okay, that's better than notch filters. Sometimes you have to use notch filters, whatever, but it's better if you can just really optimize things so that you can get rid of the noise before you record. Okay, in the UK, or like in Europe, by the way, it's 50 hertz, not 60. So it was a joke in my old lab that the gamma oscillation in American rats was like higher frequency than the gamma oscillation in European rats. Because gamma is largely artifactual. Anyway, so the other thing that's interesting, by the way, is that we have less 60 hertz noise in the Bronx, probably than a lot of places. And the reason why is because in the 70s, there was all these buildings that were getting burned down because rats would shoot through the cables and set them on fire. So then they, maybe it was even earlier in the 70s, so then they came with a stuff called BX conduit, which stands for the Bronx. And it became the housing code and everything around here, where all of the electrical wires that run through the wall, they have two wires running through the like hot and cold wire, and then they have it's shielded with this metal conduit that rats can't chew through, and that's grounded. And so that's connected to all the outlets, so all the outplates and everything. So actually all of the electrical wiring in the Kennedy building, so actually it must have been older than the 70s. So at least when they put in new wiring and stuff, it's all shielded because of rats chewing through the cables. So, so the Bronx, that's the contribution of the Bronx to neurophysiology. So, all right, important point, filtering. You can think of it as removing certain frequencies. It's more complicated than that, but for now, let's just think about it as that. Multiplication in the frequency domain, just filtering, right? Or you can conceptualize that as filtering the same as convolution in the time domain. So this is sort of the same thing. The filtering corresponds to convolution with a kernel. So we often talk about the convolution kernel, which can be a Gaussian, for example, convolution with a Gaussian, which is the same thing. So interestingly, the Fourier transform of a Gaussian is also a Gaussian. So if you convolve with a Gaussian for like your hot air balloon image, it's actually the same as taking the Fourier transform of that and then multiplying it times a Gaussian transfer function. Anyway, I don't want to get too much into that unless we have spare time later. All right, so this is one of the things I think is important is we can conceptualize convolution as filtering. So the GCamp signal is a filtered version of the spike trains, or the channel rhodopsin conductance is a filtered version of the light pulses that you give, or alternatively, a microscope image is a filtered version of the true fluorescence, true distribution of fluorescence in the sample. Right. Yeah, I guess I already said that. Any sort of distortions by an instrument like point spread function microscope, those are all convolutions. And then that's if they're operating a linear range. So GCamp has some nonlinearities, which can be important. So we'll talk about that later. Those two act as low pass filters. Okay, any questions before we move on to sampling? Could someone at least say we have no questions?
I guess I have a question.
Okay.
Oh, okay, you can hear me. Can you go back one slide? So, for things like GCamp and channel rhodopsin, thinking of it as like a distortion of the signal, is it, or is that just the signal? Like,
okay, yeah. So I think a way to think, yes, all right. So I guess my point is that you can think of something like channel rhodopsin as being a linear system, and the input to channel rhodopsin is the photons, and the output is its conductance, right? And then of course, if you measure with an electrode, then the input to your electrode is channel rhodopsin's conductance, right? But I guess what I'm saying is that the reason it's relevant to think of it as something like a distortion is that when you give a series of light pulses to a cell, the important thing to recognize is that you are not injecting the photon, the waveform of the photons that you're giving, excuse me, is not the same as the conductance waveform the cells are seeing. You see, and it's important to recognize they're not the same thing, that you think you're injecting this perfect thing, and that's not actually what channel rhodopsin is doing. And the same thing with GCamp is that what you're seeing is not actually, when you use GCamp, you're interested in the spikes. I mean, most cases you're interested in the spikes, right? So what you're measuring is not the spikes. What you're measuring is a distorted low pass filter version of the spikes. So I'm just saying it's important to recognize that disconnect and what the mathematical relationship is between those things so that you can plan your experiments correctly and take all that stuff into account. Okay. So with GCamp, they have these what are called deconvolution algorithms that essentially take what's known. I mean, there's different ways you can do it, but the idea is that we make measurements of the GCamp's on and off kinetics in vitro and so forth. And there's also different methods called maximum entropy deconvolution. So there's, for example, a method where if you know that spikes are spikes, like if you know in advance that action potentials are spike-like events, so you know that the underlying function is spiky looking, then you can optimize, you can essentially find what the optimal deconvolution kernel is that gives you a train of spikes that looks the most spiky. and then that will be an estimate in essence of what the convolution kernel is for that particular cell. So I think different cell types and stuff like that or even different individual cells in the same sample, the convolution kernel can be a little bit different because of calcium binding or who knows what. So in microscopy, people also do stuff like that you can take an image that's slightly blurry, and then you can find a way to essentially estimate the point spread function based on what is the thing that makes this image the sharpest. All right.
Okay. Yeah, I guess I was thinking particularly of like sub-threshold things that you might pick up with like voltage imaging or something like that, but that's
so
like separate.
Voltage versus Gcamp. Okay, yeah, okay, so that's what I was going to say is the Gcamp. All right, so the reading actually talks about this a little bit. So it talks about it, for example, that if you have any sort of event that occurs on a shorter time scale than the kinetics of your filter or Gcamp or whatever, then you can essentially treat it as an impulse response function. So it is true that Gcamp is not linear. There's a bunch of things that are not linear in there. So you can still use deconvolution, which is this linear approximation, but it turns out that the relationship between membrane potential of a cell and calcium concentration is not linear. And the relationship between the calcium concentration and the fluorescence of Gcamp is not linear. And actually even the relationship between the spike rates and calcium concentration is not linear, in the sense you have things like saturation. If you have a bunch of spikes, the calcium signal doesn't keep going up forever. So yes, those things are all nonlinear. So you can't measure things like subthreshold membrane potential fluctuations with Gcamp, if that's what you mean. So I think it's just a question of what it is you're trying to measure. All right, voltage imaging don't even get me started. That's a whole separate thing, and we can talk about that maybe another day. Maybe Tiago will talk about it in his imaging thing. Voltage imaging is much, much harder for a bunch of reasons. And I sort of, I remain somewhat skeptical that it's ever going to work. In like real world situations, give us something that's really, really useful. There's a lot of reasons it's very difficult. I think people are close to getting it, but it's still like the amount of effort you have to put into it and how sophisticated of a setup you need compared to the quality of the data you get is not very good right now, and probably forever. So, all right, sampling. How do you configure your filter settings and sampling rate, or it could be sampling density, or however sampling occurs? How do you do that correctly? All right, so I should say that all these slides I just stole from some other people last night. So some of them are crappy, and that's just how it is. So this is a good sampling. So if you can see there's the blue waveform underneath, the blue sine wave. Okay, and then if you sample here, it's like 8 times per, like you have 8 sample points per period of the wave. Then you can see you get this really nice reconstruction of the waveform, right? So this would be really bad. Here you're sampling it at 1.5. So if this is if this thing is sampling at like 10 kilohertz, or say this is an oscillation at 10 kilohertz, you're sampling at 15 kilohertz, then you're not getting it, and you're actually getting something that's distorted. So this is also still bad. So this would be if it's say 10 kilohertz and you're sampling at 20 kilohertz, then you get this thing that is still very bad. So the idea here, though, is that this is the bare minimum, and there's this thing called Nyquist theorem, which basically says that if you have a certain frequency present in your signal, then you have to sample at more than twice that frequency in order to capture that. So this is more what I mean. So here's if you have a sampling rate that's 3 times the frequency of the sine wave. And it's still not perfect, but you can see that you basically capture essentially that there's a sine wave at that approximate frequency. Whereas at these other things you don't. And actually there's a phenomenon called aliasing, which is that which is here where when you're undersampling. So if you are sampling at a rate that's lower than the frequency of the sinusoid, then it actually generates an artifactual sinusoid at a different frequency. Okay, so blues, blah blah blah. Yeah, so this is called aliasing because it aliases that a high frequency that's higher than half your sampling rate. into one that is lower than that. Right, so here are examples of aliasing. Here's an example of when the strobe light turns on. So if the strobe light was flashing at a much faster rate, right, then you wouldn't see this. See, it looks like it's turning slowly, so you're essentially getting the emergence of this lower frequency oscillation. So this is actually complicated because there's actually many different sources of sampling artifact and stuff here, in the sense that you're illuminating the fan with a light that oscillates at 120 hertz, and then you're imaging with a camera that probably has like 30 hertz frame rate, and then you have this, anyway, but that's the idea. So here's another example that's more subtle. I made this last night, but so you have Dracula here, which is black and white. So can everyone see there's these oscillating waves there of red, green, and blue? So what this is that I just put Dracula on my computer screen and I took a picture of it with my phone camera. And so the idea here is that in the spatial domain, that the resolution of my phone camera was not more than twice as much, so that each pixel in my phone camera was not, the pixel density was not twice as high as the pixel density in my screen. So then you actually get these low frequency or low spatial frequency oscillations that mismatches then aliased into these lower spatial frequencies you can see here. And it's also interesting that there are different colors, and that I think is because the red, green, and blue pixels are right next to each other. So it goes like red, green, blue, blue. And so you can see that they're actually, I don't know, aliased next to each other. That's another type of aliasing that you can see. Okay, so how do you avoid aliasing? So this is important. So the idea is that in order to do this, there's no, well, what you have to do is you have to put a low pass filter that's in hardware, or somehow there has to be an intrinsic low pass filtering that occurs in hardware that is prior to sampling. By sampling, I mean A to D conversion. So there's a sense in which, okay, so if you have a microscope, let me see what the next thing is. Okay, all right, so let's electrophysiology first. So active potentials, frequency content up to about 8 kilohertz, and that's just empirically been determined. So what frequency do we need to sample at? I'd actually like someone to answer that question. What do you guys think? What's the minimum frequency we would need to sample at?
16 kilohertz.
16 kilohertz, exactly, right? So at the bare minimum, we need to do 16, but in reality, we need to do actually more than that. So what I would say in practice is that when people do recordings, when they want to look at active potentials, they typically record, all right, so if you're doing, say, intracellular recordings, or some sort of extracellular recordings where you really want to look at the spike waveforms carefully, then people sample at like 40 kilohertz or 30. People do more regular sort of extracellular recordings where the spike waveforms aren't quite as important, and you sample more than 20 kilohertz. So you save disk space. But you wouldn't sample below 20. And I sort of used to do 20, now I'm kind of doing more 30. So it takes 50% more disk space. But then you do get nicer reconstructions of the waveforms, which can be helpful for things like, sorting cells into like different types of interneurons based on their spike waveforms, something like that. Right. So yeah, so that's correct. It's 16 to be the minimum, but in practice it's more like 20 to 40. Right. So so what you actually see at above 8 kilohertz is, it's not really that you see sinusoidal oscillations, which then are cleanly aliased into a different sinusoidal oscillation. It's that anything above that is just random noise. And then it gets aliased down into other random noise. So then basically low pass filtering in practice just means that if you don't have a low pass filter in there and you just recorded, then what you would just see is just a lot of extra noise that is not necessary and not helpful in any way. So one sort of example to that, so it is very useful to know what the different possible sources of noise are in your whatever data collection. So the most common thing is like 60 hertz and harmonics of 60 hertz. So most lights oscillate at 120 hertz actually. So if light bulbs or fluorescent lights, whatever, so the so the AC current looks like that, but what actually the light looks like as a function of times, it looks like this. Because you have light there, and then you have light there and light there. So it's actually this oscillation at more like 120 hertz and then harmonics. So I don't know, photometry signals, for example, or microscopy can be contaminated with things at those frequencies. The refresh rates of monitors, so that's much less true now that we have flat panel monitors. But even so, refresh rates of monitors, you can see, at certain frequencies, like 60 or 70 hertz, whatever. So it's important to know about what the different, so those are all annoying for things like local field potential recordings, because you're trying to record stuff in that range. You can't just notch filter it or whatever. So one notable exception, I'll say, that where you can get periodic noise sources that are up in kilohertz range are switching mode power supplies. Things like iPhone chargers, or anytime you have something you plug into the wall that has a low voltage DC thing that comes out, that powers an LED or a camera or something like that. Those things often generate noise around, like, say, 20, 30 kilohertz. And if we have time at the end, and if anyone's interested, maybe you're not, I can talk about why that is and how those work. But anyway, just so you should know what your noise sources are. One thing I'll say that maybe it's interesting in this context. So we have a lick detector that we got from Genelia. And if you're doing electrophysiology in a mouse that's licking a spout, you want to be able to detect when the animal licks the spout. And this is actually not a trivial problem, because you can have like a vibration detector, which works, and then you can also have infrared light beams. But if you want to pack them close together and you want it to be very sensitive, then the best way to do it is just to have it electrical and then just pass current through the animal, right? And you can pass such a small amount of current through the animal can't feel it, but that's the most sensitive way to detect a lick, is just having it pass current through the animal. But the problem is when you're doing electrophysiological recordings, where you're recording these microvolt signals, then anything is going to be a huge artifact. Certainly going to be more than microvolts. So the way that these people at Genelia fix this, so remember I was saying that we have high pass filters on low pass filters on our amplifiers around somewhere around 8 kilohertz. So what they do is that they pass current through the animal, but it's at a frequency of about 1 megahertz. So it's super high frequency oscillating current that passes through the animal from the lick spout into the animal and back through the ground electrode. But the frequency is so high that it's all low pass. That it's all filtered out by the low pass filters on the acquisition software. So anyway, that's a somewhat clever application of this. So, all right, sampling. Only have frequency content ranges. Okay, we got to use low pass filtering to remove everything. Trying to think of, sort of exceptions to that. So can't remember if I said this or not, but so maybe a way to say this is either we have to use low pass filtering, or it can be the case that a lot of times you have an instrument that's only fast enough to operate up to a certain wave or frequency. So a lot of times you might have like an amplifier, if you have an amplifier that only goes up to, I don't know, so say for example, I have these fiber photometry photodiode things that have these really slow amplifiers, and the amplifier, basically the bandwidth of the amplifier only goes up to 70 hertz, which is infuriating. So I don't need to low pass filter that, but basically the amplifier is like a low pass filter, and so I would have to sample that at more than 140 hertz. I'm sampling at 20 kilohertz because it's just more convenient to do it that way. But also a microscope, right? If you think about having a camera on a microscope, the number of pixels you need, or the frequency of like the packing of the pixels just has to be more than twice as high as the frequency cutoff in the point spread function of the microscope. So the microscope itself is acting like a low pass filter on the distribution of fluorescent molecules. And so you essentially, I guess how you have to sample that, like how many megapixels and stuff you need, depends on essentially the frequency response, spatial frequency response characteristics of the microscope. So I guess in a low pass filter would be like if you put something, so when I took a picture of Dracula, if I put some sort of lens in front of Dracula that made him a little bit blurry, then you wouldn't have seen those wavy lines. You wouldn't have seen that. It's called a Moiré pattern. So it's not necessarily that you necessarily add a low pass filter. It's just you have to, sometimes you do and other times it's sort of intrinsic to the system. Right? At least twice the frequency, but in practice, use 3 times or more. Notch filters, you can use them. It can be problematic, and it's really, and even if you're going to use a notch filter, it's really best if you invest more time up front, grounding things properly, and really trying to get the best quality recordings that you can. And I think in general, that's a general principle when people do recordings, is that in theory you can do all this stuff in software post hoc, but if you're going to record an animal with a camera, spend more time and make sure the lighting and everything is really perfect so you get a really nice clean image. It's much better to do that than it is to have sort of a crappy image and then try to computationally clean it up afterwards. And the same thing with any sort of thing, that just because you can in principle clean it up later, it's still better to spend your time optimizing at the collection end. Right. So I think we'll take a break before we move on to the next part. Does anybody have questions about the first half? I'd like to make a rule that somebody has to ask a question. I'm going to call on somebody. Victoria, you look particularly blasé.
You said I look like what?
I said you look particularly blasé today.
Oh, it's because I'm trying to read while listening and writing notes.
Okay.
I'm very interested, but I don't have any questions right now.
You're very interested. Okay. All right. Well, if really nobody has any questions, then let's take a break for 5 minutes, and then we can come back and talk about what the data looks like when it's inside your computer.
Actually, I have a small question.
Sure.
So, like in practice, the general rule is using 3 times higher frequency, but
Yeah.
how, like that varies depending on your application, right? So how would
Right.
you figure out like the exact range that you need?
Okay, yeah, so that's actually a good question. So, and something I was going to talk about but didn't. So I guess the first thing to do is figure out what is the signal you're trying to measure, and what frequencies are present in that actual signal, right? So I guess that's why I was saying that people have done measurements of action potentials and found that action potentials, don't occur on megahertz. There's no nanosecond stuff going on in an action potential. The ion channels have a certain rate that they open and close at. So that's, people have measured and determined that action potentials don't really have any frequencies above 8 kilohertz. So if we know that knowledge in advance, then we can say, well, we don't need to sample at higher than that. So we can put a low pass filter at, say, 8 kilohertz. But we're picking that 8 kilohertz or 10 kilohertz or whatever cutoff based on our prior knowledge of what action potentials, like what we expect action potentials to have, right? What frequencies we expect them to have. And then we sample based on that. So another thing also, here's a counterexample, that we wanted to put these, so mice make these ultrasonic vocalizations. When they're like happy or sad or whatever. And so we wanted to get a microphone for that. So regular microphones, so a CD player, which is like the standard formats are based on the digital formats for CDs. I believe they sample at like 44.1 kilohertz. So what that means is that that's the sampling rate of digital audio formats like WAV and MP3 and all that. So at least by default. So what that means is that the highest frequencies they capture are something like 22 kilohertz. So but mice, so for humans, that's like the top of human hearing, but mice can hear in ultrasonic frequencies. So mice make these, whatever, calls at something, I forget exactly the frequencies, but it might be like 50 kilohertz or something like that. So what that means is that we have to have our low pass filter set, higher than the highest frequency sounds that a mouse is capable of making. And then we need to sample at a multiple 3 of that. So, so for example, our electrophysiology setup samples at 20 or 30 kilohertz, but mice are making these things at like 50 kilohertz. So we had to buy this special microphone that samples at something like 220 kilohertz. And has low pass filters, probably somewhere around like, I don't know, 90 or 80 kilohertz or something like that. And then we'll have to sample those things separately and then synchronize them post hoc. We'll have to synchronize our electrophysiology, our video, our behavior, and this microphone, because they're all in different systems with different sampling rates. Okay, so or behavior maybe is another example where you can use a 30 hertz camera, because the position of an animal's body changes, and positions of an animal's body does not change on millisecond time scales. It changes on maybe tens of milliseconds time scales. So I guess the key answer is that you have to know what it is you're trying to measure first and what you expect to see, and then base it on that.
Okay, thanks.
Sure.
I have a question.
Yeah.
I feel bad asking questions because I didn't do the reading. I'm sorry. So
Don't
just
feel
let me
bad.
know if it's just let me know if it's in the reading, and I'll find the answer. But
You can ask questions even if it's in the reading. All right, that's really okay.
okay. I'm trying to like squeeze in some last experiments.
You should stop
anyway.
apologizing so much, Claire. We've talked about this before. All right, anyway,
Okay.
yeah.
Sorry for apologizing.
There you go.
No, stop.
Okay, so you mentioned that you need to have some sort of like in the hardware a low pass filter
Yes.
even before you're collecting your signal.
Yes.
What order is that on? Like, what are you filtering out?
So
the
so
reason
what do
I
you-
ask
Yeah.
is okay, so I'm recording and I'm collecting both the spikes, but then I also have the LFP information.
Right.
So in order to capture that, I'm just wondering, like, at what level am I filtering out?
So
while I'm
you're
collecting
using the
it?
Intan preamps, right? So I forget the
Yeah.
I mean, it's all the specifications on their website. So the Intan preamps, they actually bandpass filter the signal. So they low pass filter out. They low pass. So the low pass filter filters out everything above something like 8 kilohertz. I don't remember the exact number. And then the low pass filter goes down to something like 1 hertz or 0.1 hertz or something like that.
Okay.
Or I'm sorry, the high pass filter.
Yeah. Okay. I was wondering because I like get both signals at once, so it seems like it would
Right.
be bandpass in this case.
Well, so yeah, I mean, so the reason that people often you have to do high pass filtering with stuff like electrophysiology is more a question of dynamic range. Let's talk about that in the electrophysiology lecture. I just keep building on these principles, but the idea is that there's no physiological signal at above 8 kilohertz, right? So, but there is noise above 8 kilohertz. If you just have an electrode, thermal vibrations and whatever, there's noise at these higher frequencies. So if you if you're not low pass filtering, then you're just sampling all that noise, and that high frequency noise is getting aliased down into the frequency ranges of the signals that you're trying to record.
Okay.
So I mean, the thing is, stuff like that, the equipment you buy typically has all this stuff built in, right? Because so that's what I'm saying. Like the Intan preamp has it built in. So, but you should still understand it because I mean, you still need to understand this stuff. All right. That was
Okay.
your fault, Claire, that I just did that. So,
Okay.
all right.
I have a technical question.
Yeah.
Can you add me to whatever list everyone's on that gives them the Zoom links?
Sure.
Thank you.
All right. Well, let's take a break and 5 minutes or something. All right. Okay, everyone, maybe we should get started again. Are people back? Close? Mostly. All right. All right, so one thing I should have mentioned before, just to be clear about terminology and stuff. So, so Fourier series it's like this. Well, don't worry about that. That's when you have like a periodic function, then you can represent it as a sum of sinusoids. So the Fourier transform that people talk about, and if we have time, maybe we'll talk more about it at the end, but that's like the specific thing for taking a function that's usually not periodic, like a signal. So when it's, well, I won't get into that, but it's some sort of actual piece of data or something, some function you have, and then converting into its frequency domain representation. So the fast Fourier transform, or the FFT, so that's the name of a specific algorithm for performing Fourier transforms. And so Fourier transforms historically were very computationally intensive to compute. And then someone, I forget who it is, this huge breakthrough in computer science, they came up with the FFT, the fast Fourier transform, which allows you to, it's like order n log n instead of order n squared, where n is the number of like points in your, like in the number of elements in your vector. And the way that it works is that it generally only works on if you have like your data set is a power of 2, in the sense it has to be like 1024 elements long. It can't be 1025 elements. And the reason why is that the way it works is that it takes your data set and then it, like your signal, and then it cuts it in half, and then cuts each half in half, and then each of those halves in half all the way down. And so even if you had like 1025 data points, then you would just add zeros until it was 2048 long, and then you would FFT that. So it actually turns out that the newer commercial FFT implementations will actually look for multiples of other, or powers of other small integers. So it might divide it into thirds, and then each third into halves and whatever. But anyway, but that's, so just, that's this whole notion of FFT as an algorithm, and why it is that, the length of it being powers of 2 is relevant. I say that largely so you can look smart later on and not look stupid. So I think that's an important thing to pass on to the next generation of scientists, is making you not look stupid in the future if someone talks about this. So, okay. Variable types in digital formats. So what does your data look like? All right, so the first thing is integers. So like I said, typically data acquisition, that the A to D converter will return essentially, an integer that's saved on your hard disk. An integer meaning, so if you have an 8-bit A to D converter, then what happens is that it'll take that analog voltage and will map it onto something that's 8 bits like this, where you'll have 8 zeros and ones in a row on the hard disk or in RAM or whatever. And so it can either be an int 8 or a uint 8. So these would be the names of the types of the variable types in your programming language. So and they're not completely standardized between different languages, but for most, like Python and C and MATLAB and so forth, you have like, an int 8 variable type means that it's 8 bits. And that means that it can store a number between negative 128 and positive 127, which is essentially half of 2 to the 8th. So a uint 8, an unsigned int 8, goes between 0 and 255, which is 2 to the 8th minus 1. So I think we talked about this a little bit last time. So the difference here is that the signed integer, it actually only has 7 bits to store the value, and then the last bit stores whether it's positive or negative. So in both cases, the thing can take, this thing can have 256 possible values. It's just a question of the range, whether it's something that is impossible to take a negative value, or whether it's something that can take a negative value. And same thing for int 16. So this would be, for like if you had a 16-bit A to D converter, and 32 and 64 and so forth. So it's rare that you'll see more than like a 16-bit A to D converter. It's usually, so 8-bit A to D conversion is probably most common in cameras, where if you have an image, like an RGB image, often it is something where, so they'll say 24 bits per pixel, most common image formats. What that means is that there's 8 bits for red, 8 for green, and 8 for blue. So that at each individual point in the data, there's a value, and it's usually int 8. So it's uint 8, where it's a value between 0 and 255 for how much red is in that pixel, how much green, and how much blue. Whereas other stuff, usually it's higher, more than that for electrophysiology and stuff. Okay, so there's a couple limitations of this. So also, I didn't make any of these slides. It's just, I should credit the person who did, but I don't know. Anyway, so these fixed point integer numbers, right? So it's simple, it's exact. The range is too small. That's the big problem. And you can encode numbers that are not integers, like 2.5. So other issues. So overflow. So that's if you have a number that goes from 0 to 255, and if you have a number and then say it's 255, and then you add 3 to that, then the answer will actually be 2. Because it will go 255 plus 1 is 0, plus 2 is 1, and plus 3 is 2. So it loops back around. So that can be a problem, but sometimes it's actually desired behavior depending on your application. But a lot of times you don't want that, so you need to use more bits. So this thing, it says in MATLAB that if you have an int. If you have an int 8 that's at 127 and then you add 1 to it, that it'll just keep it at 127. What MATLAB actually does is it'll then say x equals 128, and then when you go back and look, now all of a sudden x is a double and not an int 8. So it'll simplistically convert it to type double. So the double meaning double precision float is the standard variable type in MATLAB, and it's standard variable type for most like NumPy and that sort of stuff. Okay, truncation. Yeah, so obviously you can't have a non-integer value for things that are encoded as integers. So what this means in practice is that you can't do any math on, or it's very difficult. You can like add and subtract stuff, but you can't do any sort of real life math on something that's stored as an integer. So if you get around that, use floating point variables. So floating point, it's exactly like scientific notation, where forget all that crap. Anyway, I'll show you in the next slide. So data is typically acquired as ints, but then you have to use floats for any sort of actual, analysis that you do. Now, and this is another sort of point, is that CPUs do integer and floating point both. So floating point and integer calculations are different and actually done by different parts of the chip. And so GPUs are these specialized things that only do floating point operations, and they only do sort of specific types of like matrix floating point operations. So they're much, much faster. So in the old days, Intel made a chip they made the 386 and 486 series in the 1990s. And they had the 486SX and the 486DX. I'm assuming none of you are old enough to remember this. And so the 486DX had what's called a math coprocessor, which is that there was a separate chip on the motherboard that just did nothing but floating point calculations, so that could be fast. And so back in the 90s, people had this thing where they had the CPU and then they had the math coprocessor, and they would run these neural networks on there. They thought they were like cutting edge, hot shit back then. And then all that stuff died out, and there's no math coprocessors anymore, and nobody was doing neural networks. And now we have GPUs and we have all this deep learning stuff, and it's exactly the same thing, just version 2.0. So it's kind of funny to me that the math coprocessor has come back, but they call it the GPU, and it's supposed to be really cool. Anyway, GPUs are great though. They're much more powerful than math coprocessors. Anyway, so here they talk about real number representations. So reals are, some languages call them reals. I just call them floats. The only language I've ever really encountered that calls them reals is Pascal, which, Pascal, whatever. We're not going to talk too much about that. But instead of here, where you have just the 8 bits that encode, the number and then the sign. So for here you have, so there are no unsigned floats. It's like you have one for sign, and then it's encode the exponent, and then that's the actual sort of number. Like this, you could think of like the significant figures, like sig figs, remember from high school. Same sort of thing here. So a single is 32 bits, and a double is 64 bits. Okay, so if you remember that an int 8, for example, is 8 bits, so that means that the smallest floating point variable you have is 4 times that size. So these are what we need to use for any sort of math, but they take up much more memory, right? So this is one noteworthy thing that I'm just bashing Pascal, but it turns out that you make a lot more money as a Pascal programmer apparently in industry if you want to leave. You might note that Python is all the way down there. MATLAB's not even on the list. MATLAB would be like -50%. So something to keep in mind if you leave academia. All right, so fixed it now, calling it floats. So single and double precision floats. So in MATLAB, for example, they're called singles and doubles. You have precision error, right? Because so I guess with integers, the issue is that you can only encode a certain range of numbers. You can't get numbers outside that range. With floating point variables, you can encode extremely large or small numbers, there are some sort of limits, but you almost never encounter them. But you only can store essentially a certain number of decimal points of precision. So that's where the limitation comes in. So you can store huge numbers to give a certain amount of precision. So the reason we have single and double precision is because there's actually some calculations where single precision will not be precise enough. And particular things like if you have error that accumulates, like sort of numerical integration that's integrating every step and error builds up. So in practice, double precision is almost always good enough. And it's one of those things where you just use doubles and don't think about it, is what I generally do and what everyone else generally does. I want to give a real world example of this that I'm very happy about. So I think it really illustrates the point. Okay. So it illustrates the point. So there's this famous theorem, Fermat's last theorem. What it says is that there's no positive integer solutions to this equation for n larger than 2. So if n is 1 or 2, there's an infinite number. But if n is 3, 4, 5, if n is an integer, then there's no integers x, y, and z where this equation will be true. And this took hundreds of years for people to prove, and it was thought for a long time that it was considered this unprovable theorem of mathematics. And there's like Star Trek episodes where they show in the 24th century people, bemoaning the fact that this theorem was never proved. So this guy Andrew Wiles proved it in the 90s, and it was considered this greatest mathematical achievement of all time. So he was too old to win the Fields Medal, so they invented a new Fields Medal called the Fields Medal of Distinction to give to him because it was the biggest achievement in the history of mathematics. So, however, so there's a Simpsons episode, okay? So Homer finds out that he inserted a crayon into his nose as a child and it's stuck in his brain. So then they remove the crayon and then Homer becomes really smart. And so Homer proves his donut theorem on this board here, right? And if you look closely, you'll notice something, which is that. The question is Homer correct? Is this actually true? Now, so did Homer Simpson actually disprove this? So is that actually correct? Does that actually hold up? So if you take a scientific calculator and you type in that equation, this plus this minus that, do you get an answer of 0? And the answer is yes, the answer is 0. Homer Simpson has disproved Fermat's last theorem, right? So can anybody speculate as to what might be going on here?
It's a rounding error because of the limitations on the hardware?
Exactly, because numerical precision, right? Okay, so let's see what happens. Oh, well, and that's what's relevant. So that was the outcome. But then what happens if we use double precision floats? All right, so. Okay, so did Homer, can everybody see this okay? Yeah, all right. All right, so now, okay, so set up the variables. So this is what the theorem says. That's what Homer wrote, okay? So that's X, Y, and Z we have here. And if you look here, they're all doubles. They all take 8 bytes, which is 64 bits of memory. Okay, so we're using double precision floats now for Homer's calculation. The answer is yes, that x to the 12 plus y to the 12 equals that, z to the 12 equals that. And if you look at that, they are 1, 2. For the first 10 decimal points, they're the same. Okay, so that's why on a desk calculator, the answer is 0, or on a scientific calculator. And that was the writers of the show designed it specifically that way. So, which is one of these things when I actually learned about this, I was like, that's on The Simpsons? That was more sophisticated than I thought they were. And so the question is here, using double precision floats, how large is the error here? So the error is 1.2 times 10 to the 33rd. So it's not like it's off by 6. It's off by a gigantic amount. But single precision arithmetic will tell you that it's 0. So numerical precision. So doing math on a computer is not the same as doing math in real life, or doing math analytically, right? Anyway, so like I said, you're mostly better off just use doubles and don't worry about it. So the problem is when you get into trouble with that. Well, there's a couple limitations, I guess, of that. So one of them is memory. One of them I'll say actually before that is the GPUs are optimized for single precision floating point arithmetic. And the reason why is because computer graphics, it's like a close enough thing. Like if the image is rendered correctly, you don't need double precision math for that. So GPUs are highly optimized, but they're optimized for single precision arithmetic. So if you can do your GPU calculations with singles, then you get this full speed benefit. So when GPU math first started coming out and being popular with CUDA and all that, then there was no double precision support at all. And then they added double precision support later. But the double precision support is more than twice as slow. So in some GPUs, it's like 20 times slower, and in some it's like 3 times slower or something. So you actually have to look into that. I think the newer ones probably is better, but this is something that was a problem for people. But I mean, this is a real life example of something where they use single precision because it's faster, but in theory it should only be maybe twice as fast, but because it was highly optimized for that, then it's worse than that. Right, so the other thing is just a question of memory. So let's go back to MATLAB here. So I'm going to load in some data. This is fake data. All right, so and the number of channels. So you can see here that this is the data reshaped. Here so this is data that is fake data from the Intent acquisition system. So we have 7 channels, analog data, and it's 219,600 data points. The sample of 20 kilohertz, so it's somewhere around like 12 seconds of data. Right, you can see it takes up 3 megabytes of RAM. Okay, so this is only 12 seconds of data. It takes up 3 megs of RAM, and that's in16. That's the original format. So what if we want to do, for example, z-score that or something? So so if we were going to do any sort of math, we have to change it to doubles. And then now, so I made the double version. So now the double version, which we can actually use for mathematical operations, is now 12 megabytes. I guess that's for 12 seconds of data. So if we had 1,000 seconds of data, or I'm sorry, 12,000 seconds of data, right? Then this would be 3 gigabytes and 12 gigabytes, right? So now all of a sudden your 3 gigabytes of data takes up 12 gigabytes of RAM, right? So what if we had 50,000 seconds of data, right? Then it starts to get bigger than what fits in the RAM on your computer. So this is just something you have to be aware of and think about how to do this carefully and working with your RAM and all that. Because it's something where you really, I mean, I don't think I've ever used singles for anything ever. So, I mean, but you just have to be smart about how you handle your data, knowing that it's going to get blown up a lot and use a lot more resources than what you might think. Any questions before I move on?
What do you do in your lab concretely for that?
For what?
When you, how do you save? Do you save on external hard drive? Do you save online? Like what type
That's
of?
actually what we're going to get to next, is more sort of concrete strategies for data storage and stuff like that. So, what time is it actually? Yeah, so that's sort of what we're going to talk about next. All right, so I mean, yeah, just how you deal with it. I mean, so the way you would have to deal with stuff like this is that you can only load in chunks of data at a time that will fit in RAM, and then deal with them. And so if we're doing things like local field potentials, you actually would downsample. I don't think we actually, maybe we'll talk about that in the EFIS thing. So that, the spike analysis doesn't require loading all that stuff in memory. It's actually, we'll talk about when we get to the EFIS stuff. There are hoops that people have had to jump through to figure out how to do this, because your whole data set will not fit in RAM. Right, so there's lots of hoops to jump through for that. Usually you don't have to deal with them. Usually it's like the people who made the software that does the spike sorting, they had to deal with it, and then you don't have to. All right, so how much memory from variable types take? So 8 times as much, is that right? Sorry, was that right? 4 times as much. Yeah, because it's an int16. So if it was an int8, so imagine an image, right? If you had an image that's 8-bit, 8 bits per pixel per color, right? So then it's actually a factor of it's a factor of 8. So like, you know, I don't know. So 2 photon imaging data, if that's 8-bit, then and that's already huge data sets, and all of a sudden you're taking eight times as much RAM than the actual image has information in it. So. Actually, come to think of it, if you had 8-bit acquisition, is there any reason not to use single precision floats? Probably not. You know, anyway. All right, so how is memory and storage organized in a computer? What am I talking about next? Okay, yeah, so this is all just, I don't have any slides for this or anything. So the first question is, how big should I expect the data file to be? All right, so I think we talked about this a little before, but this is something you have to think about, and it's not complicated. So, well, what I should say is that, okay, so there's compression, but compression takes time. And usually it's the case that when you acquire some sort of data from something, it gets written in some sort of relatively simple flat binary format, which is the raw output of the A to D converter. And so, so I guess what I'm saying is that if you have, all right, so let's say that we were doing, say we were doing 256 channel recordings, 256 channels, and then let's say 20 kilohertz. And it's 16-bit A to D conversion. So I'll actually just say 2 bytes, right? And then say we record, you know, I don't know, 100 seconds of data or something, right? So then that is, let's see, 256 times 20,000 times. So that would be 1.02 gigabytes. All right. Yeah. Well, approximately, right? So gigabytes. So that there's actually not, there's not a million, there's not actually a billion bytes in a gigabyte. It's, you know, whatever. So there's 1,024 bytes in a kilobyte, not 1,000. So yeah, just forgetting about that. So you have about a gigabyte. Okay. So but this is the kind of thing you should be aware of, and you should think about. You should be like, well, how big of a hard disk am I going to need? How much space do I need before I start this experiment? You know, if that's 100 seconds and you're going to record for, you know, 10,000 seconds or whatever, then you need to have 100 gigabytes free and ready to go to store that. Else your experiment's going to crash in the middle. So that's one thing. And this is, I mean, fairly straightforward. Does anyone have questions about this? Does this make sense to people? Okay, so that's one thing, is that part of the reason I'm trying to say this is I know it's not that complicated, but that's actually the point, is that it's not that complicated, that this is something that can be demystified, and it's actually not that hard, right? So you can look at the size of your data files, and it will often be exactly that. Like you can look and be like, oh, that recording is 93 seconds long, and then you look and it was exactly 93 seconds long. Right? So what else are we going to talk about? Okay. So maybe let's talk a little bit about compression. So, right, so with compression, there is lossy and there's non-lossy compression. Can someone tell me what the difference is between those things? I know someone can. Can someone please tell me?
Is it when you lose data? Well, lossy is when compression causes the loss of information.
Right, exactly. So that it stores an approximation of the original data, but it's not 100%. It's not perfect. It's not a perfect reconstruction, right?
Mm-hmm.
So what's an example of a lossy compression format that's used just like for consumer stuff?
JPEG.
Right, JPEG would be a good example. What's another one for audio?
MP3.
Good, MP3. So all of the video codecs that people use are lossy. Just so you know, I mean, and so lossy ones, the amount of compression you get is much, you get like 10 or 100-fold better compression ratios. And it's also something you can control where you can make sort of a higher quality file. So you can make a really small video file that just looks bad, or you can make a really big one that looks good, but it's like a parameter that you can adjust how much compression, how much loss you want to have. So non-lossy compression formats would be something like PNG. So that I most would commonly use for is sort of a substitute for JPEG. For MP3s, what is it called again? I don't know. I forget the name of that. There's a lossless one that people use sometimes. Well, I should say that uncompressed. Uncompressed in this case for audio would be a WAV file. And then there are blanking on the name of that one. It's like LL, I don't know, whatever. But there's a lossless one of those. Uncompressed video, or uncompressed, I should say this is images, is often TIFF. So TIFF can be compressed or uncompressed. So TIFF can either be uncompressed or lossy or non-lossy compressed. So they call like LZW compression algorithm. So the codec, so these things are like the name of what they call their format. So what's called codecs, compressor, decompressor, that compresses these sorts of things. So and I guess for video we have whatever the big video things, H2, is it H264? You know, these sorts of things. They have these container formats like AVI, you know, MPEG, whatever, stuff like that. Okay, so uncompressed files are often huge, right? And so how hard it is to compress a file depends on how much redundancy and empty space and stuff there is in it. So if you had like a video of just total static, right? Then, or if you just had images that are completely random, JPEG, you can't, they're essentially impossible to compress because there's no sort of non-randomness for it to find and compress. But if you have some things like natural scenes, so the way the JPEG works is that it does a discrete cosine transform, which is like a Fourier transform except it's real valued. And then they only store the coefficients for the sinusoids that have the highest magnitudes, and then they discard all the low magnitude ones. And because natural scenes have like low spatial frequencies across objects and high spatial frequencies at boundaries and stuff. So so only some spatial frequencies are relevant in natural images. So you can discard all those other spatial frequencies and you can still get a really good reconstruction with a huge compression rate. Whereas PNG uses some sort of more, you know, zip type algorithm. But how well you can compress it. So with things like video compresses really well because there's a huge amount of redundancy in the sense that each frame looks a lot like the last frame and the next frame looks a lot like that one. So there's a huge amount of storage wasted in a sense by representing each, or like in a microscopy image, a lot of it is just black space, right? So there's a lot of, so I guess what I'm trying to say is that most of our data formats are actually very easy to compress or that they compress very favorably, right? That you can get large compression ratios. So there's always something to think about, you know, to use compression because, and it's also happening more and more that our data formats just getting gigantic, right? And it's always a problem for how we handle it. Any thoughts about that? So one thing I should say about compression actually is the compression is usually designed to be asymmetric in the sense that compressing something takes much longer than uncompressing it. So for example, generating a movie file, it takes, like for a 1 hour movie, it might take 3 hours on your laptop or something. And your CPUs are all pegged and your fans running like crazy. So it takes a lot of computational resources, but then playing the video file back takes very little. So that's the way that all these things are kind of designed. So you can access your data more quickly, but then saving it the first time takes a lot. And actually sometimes using compressed data formats is actually faster than uncompressed data formats. We'll talk about that next. How could that be that uncompressed data is faster, or compressed data is faster? So the idea behind computers is that when you run something on a computer, that it's only as fast as the slowest link. So broadly speaking, there's maybe two types of jobs. You could have one, well, speaking very simplistically, you could have one where it's CPU limited, which would be something like I mentioned, that you know, transcoding or encoding a video file. That's something where it uses a ton of CPU resources. And so that's something where even, so I guess what I'm saying is something could be CPU limited or it could be disk IO limited. So something like I don't know, filtering raw spike train files where you're having to read in like terabytes of data and then just, or say reading terabytes of data and then just subtracting the mean or something like that, right? So the CPU is going to be like 5%. Your hard disk is going to be spinning like crazy. And that's something where if you have a faster, if your hard disk is twice as fast, then the task is going to take half as long. So so you have to know what kind of tasks you're performing and then you know, figure out how to optimize your system for that. So so different types of disk storage or storage in general. So you have like a CPU, and your CPU is connected to RAM, and RAM is very fast. RAM is extremely fast, okay? Then and RAM and what we consider disk storage are treated separately at the operating system level. And RAM is also volatile, so if your computer shuts off, you lose it. So then you can think of also your CPU, which is also connected to disk storage, which is sort of two things, which is SSD, a solid state drive, or it can be a hard disk, which I mean like a spinning platter hard disk. And I guess you could have things sort of like tape backup, for example. Okay, so if you think about we're to plot some sort of plot where we said speed versus cost, like cost per terabytes or whatever. Let's see, so RAM is very expensive. It's very expensive, but it's also very fast. Okay, and like tape is something where it's very cheap and it's very slow. And then I guess you have stuff like, I guess it's sort of like this, hard disk, right? So the idea is you have to think about, you know, if you're doing calculations on something, you load your data into RAM and you run the calculations on it generally, right? And because RAM is fast and that works quickly. So then you have problems, for example, where if your data doesn't fit into RAM, you can try to load it in chunks or use different kind of tricks. So the spike sorting software we'll be using in a few days, or the way that actually works is that it basically requires you to use an SSD, because when you say click on something, it will have to pull it directly off the SSD because it just doesn't fit into RAM. So SSDs are much faster than spinning platter hard disks. They're also a lot more expensive per gigabyte, right? And things like spinning platter hard disks are fast enough for they're fast enough for most data acquisition. So this is another thing you have to think about. So we talk about the size of a file, right? Like, you know, how long will 1 hour of data be? But you also have to think about what's the speed of data per second. So if you're recording 256 channels at 20 kilohertz, at 2 bytes, whatever, then how many megabytes per second or whatever need to be written to a hard disk, and will my computer and hard disk be fast enough to capture that? So for stuff like electrophysiology, it pretty much is. Even with, you know, multiple neuropixels probes, you have like different drives per probe. With stuff like imaging, high-speed imaging data, you start to get to the point where hard disks are physically not fast enough, and then you need to use things like SSDs, or even striped arrays of SSDs. So there's ways to get improved performance. You can use this thing called RAID. All you should know about. Okay, RAID. It's redundant array of independent disks. That's a system where you have like a box that you put, say, 8 hard drives in, or 4 hard drives or something. And then it will, for example, store, well, actually the simplest RAID would be something you put 2 hard drives in. And then each hard drive it's an identical copy of the other one. So it's approximately twice as fast, because when you like send data to it, then it puts half the data on one and half data on the other. And so writing maybe isn't twice as fast as a copy. When you read data, then it'll pull half the data off one and half the data off the other, and your performance is twice as fast. But your write performance is also faster too, because it, you know, goes and then copies onto both. So so that's a way to get faster performance from things like hard drives. Sometimes you even need to use, so that's called striping, where the data is split across multiple drives like that. And so sometimes you need to even use striping for SSDs if you're using a high-speed imaging camera. Excuse me, I'm gonna lose my voice now. So now Intel's come out with this new thing. I want to say it's called like Optane or something, which is they have some new type of memory that's kind of between RAM and an SSD, where its speed is closer to RAM, but it's non-volatile like an SSD. And so, you know, operating systems, those things are totally separate. Like you have your RAM and you have your SSD or your disk storage, but this new thing is kind of intermediate, and I think they're going to have to start figuring out ways to take advantage of that. And probably the day is coming when all of this, when like RAM and SSD merges, and you just have like your hard disk and your RAM are just one, you know, and then other stuff like spinning disk hard drives, and so tape is something that's only used for backups. Like you put stuff on tapes and then you put them in a warehouse somewhere. And it's extremely slow, but tape is cheap and it lasts for a very long time. So this is what you could think of as what's called like cold storage versus hot storage. So hot storage would be that you're storing something and you actually need access to it, you know, on some reasonable time scale. Whereas cold storage is sort of thing where it's like you take the way most data analysis works in biological science is that you have some sort of instrument that collects huge data sets, and then you process it somehow and you extract the key elements out of it, and then now you have your extracted data that you want, and then you rarely would go back to the real full data set. So that's true for, you know, like in imaging or something. Say 2-photon imaging, you take all these huge stacks of images and then you extract the calcium traces and locations of each cell. So now that you have the calcium trace and location of each cell, you know, you can take that original full data set, which is orders of magnitude larger, and just put it on a tape and put it somewhere, right? And you're not going to have to access that probably ever again, or maybe you will, but so that's called like cold storage. So stuff like tapes are good for that. Spinning disk hard drives are sort of more intermediate, but that's the kind of thing where for hot storage. So let's see. So the other thing, so the most important thing for you to remember about today's lecture, so what's the failure rate of hard drives? I know I've asked some of you this before. What's the failure rate of hard drives? 100%, that is correct. Hard drives have a failure rate of 100%. They're like light bulbs, okay? So I'm just saying, do not ever trust a hard drive. If anybody in my lab were to lose data because a hard drive crashed, I would have no sympathy for them at all, because you know it's gonna happen. It's just gonna happen. So you have to do backups or RAID, which is redundant, where you can have a box that has several disks. So say it has 4 disks inside, and if any one of them fails, you just replace. So it has 4 disks, but it only stores 3 disks worth of data. And then if any one of those fails, you pull the failed one out and you put the new one in, and then it recopies, so that if you lose 2 of them at the same time, for example, then you lose everything. But if you lose only 1, then you can always replace it. So, all right, so you have to use RAID configurations and backups for hard disks. Okay, so hard disks are cheap. Okay, so let's talk a little bit about.
Can I ask quickly about tape?
Yeah.
I don't think I've ever heard of that before. Is like real tape, like VHS kind of tape?
They're smaller than that. It's a huge pain in the ass. Like so the way that this stuff should work is that the institution should have some sort of tape archive somewhere over the network that you would, you know, put your data in some sort of queue, and then there'd be some person there who copies it to a tape and stores it somewhere. That's the way it should work. The way that it actually works is that they have, all right, so within a lab, the best way to do this sort of stuff is what's called a NAS. It's network attached storage. So that's a storage that has like a bunch of hard disks in it. Those are in a RAID configuration. Sorry, Illustrator crashed. Okay, so the new MacBook Airs just came out, and they have quad-core i7s. And so I was debating just taking a glass of orange juice and pouring it into my laptop so that I could get one of those and have the lab pay for it. So, all right. We'll fix this. All right, so all right, so they have a bunch of hard disks in a RAID configuration, and then they're attached over the network through Ethernet, right? So that they're connected to your computer. And so then in my lab, the way it works, and I think it's the best way to do, is that any machine, data acquisition machines have hard drives, but no other machines have hard drives at all. All the hard drives are in the NOS, right? And so the reason for that is that when you run data acquisition, so a network connection, the connection over the network is never 100% reliable. So you don't want to be streaming your recordings in the middle of an experiment to a different computer. And then if that thing hiccups in the middle of the experiment, your whole thing crashes. So you need your disk storage for an experiment to be local. But then as soon as that's done, it needs to be copied to the NOS because the hard disks can't be trusted. So hard disks should not be in individual computers. They should all be in a NOS. Now that we have 10 gigabit Ethernet, so 10 gigabit is faster than eSATA or USB 3.0. So it's actually just as fast to access a NOS over 10 gigabit Ethernet as it is to access a local drive. Not an SSD. So SSDs are faster, and people should have local SSDs for sorry, I don't know why that's not stopped. Let's see. Okay. Anyway, let's see this again.
I have a question.
Yeah.
What's the total capacity for NAS then? Is it is there like a boundary? Like
So
range?
yeah, I mean, so it depends how many disks are in it, right? And how they're configured. So the probably the best configuration to use for NOSes is something called RAID 10. So RAID 10 is something where so the way that so there's different RAID levels, and RAID 10 is the way that RAID 10 works is that right, so I'll tell you a couple of things about hard disks. So they have 100% failure rate. That's number one. So number two, number three is that hard disks have gotten bigger and bigger over the years, but they haven't gotten any faster, or they haven't gotten much faster. And then their error rates have not changed much. So they have this sort of thing where there'll be like one random, bit that's wrong every 60 gigabytes or something. I don't know the exact number, but that number stayed largely constant over the years. And the speed at which they read and write data has stayed roughly constant or not much faster. So what that means is that in reality, in a sense, hard drives have actually gotten slower and slower, in the sense that the amount of time it takes you to read 1% of that hard drive's capacity has gotten longer and longer. So if you wanted to copy, so say the amount of time it would take to copy the contents of an entire hard drive, that has gotten longer and longer and longer. So now if you have a situation where you have to replace a disk, it can sometimes take like a week for the whole system to copy everything back onto that disk. So it's if it's like a RAID 5 or RAID 6 configuration where the data is distributed across multiple disks. So RAID 5 configuration, or sorry, RAID 10 is probably the best configuration. The way that works is that you have, say you have like, all right, so the way that works is that you have a thing where it's like hard disk, I don't know, I'll call it 1A, then a hard disk 1B. It's a RAID 10. Okay, and those two are connected, and both of these two are exact copies of each other. Okay, and they're in pairs. And then you essentially have like hard disk, I guess 2A, and hard disk 2B. And then 3A and 3B. So the way it works is that if each of these was 10 terabytes, then if you had a system like that, that would have 30 terabytes of storage. You see what I'm saying? So if each of these, so you have 60 terabytes worth of hard disks in there, but because there's this one-to-one redundancy, then you have only 30 terabytes of storage. But an advantage of this is that this will be also twice as fast, because if you want to read data off of these, then it can read half the data off this one and half off this one. And so it's faster than individual hard disks. And then what's nice about this is that if individual hard drives fail, so if both of those drives failed simultaneously, then you would actually lose the entire contents of the whole array. Because if you take a file, it might write like a third of the file here, a third of it there, and a third of it there. So if you lose these two simultaneously, you lose the whole thing. because every single file on there is distributed across all three.
Makes sense.
But the probability of two failing at the same time is relatively low. So you still have to have backups. But so the idea is that so that's like a NOS, which is what you have in your, in your lab. And then what the institution has was called a SAN, which is a storage area network, which is basically just a giant NOS. And what it is that they have, I think, many like clusters like this, and then those things are all connected to each other. And then there's some computer where it looks like one giant virtual hard drive, but in reality, it's tons of different boxes and everything that somehow link together. And there's their capacity for the institutional one Einstein, I want to say it's 5 petabytes or something. So it's quite a bit. The problem in the Kennedy building we have is that we do not yet have high speed. We have like a 1 gigabit connection to that building that has the SAN in it or whatever. So they actually ran optical fibers. I think it's going to be like 2 40 gigabit connections or something. Optical fiber connections. And we're getting 10 gigabit run throughout all of Kennedy. So eventually they'll want it to be that everybody just works directly off that. So I should say that when I came to Einstein, I had it negotiated and put in my offer letter that my lab would have to have 10 gigabit Ethernet, because I came to Kennedy and I was like, no, I know these cables are like 50 years old. And so it became this whole thing where then they couldn't do it, and Cameron had to go to the dean and be like, but it's in his contract, we're obligated to get him 10 gigabit Ethernet. So anyway, now the whole building's getting it, so that's gonna be good. All right, so so let's talk a little bit about how you would configure a system for data acquisition. Oh, we're actually almost out of time. So we could talk about how to configure a system for data acquisition. What else? Did I have anything else I was going to say? 5 minutes. Do you want to talk about complex exponentials? Do you want to talk about linear versus switching mode power regulators? Or do you want to talk about how to configure a system for data acquisition?
How to configure a system.
Yeah.
Okay, that's kind of what I thought you wanted to talk about. All right, so so a hard disk has a certain amount of space on it, right? And that's the physical drive, and then it has partitions inside. So as these different, you have to partition it into these different partitions. So each partition, so this is just on a single hard disk, right? Each partition, so in Windows, they're given a drive letter. Like this will be C, and then that'll be D or whatever D, right? And so and these are logically distinct things in the sense that if you fill up C, then D is not full. Okay, so so what I recommend for people to do okay, and then you can also have like a second hard drive, or maybe you could have just one giant partition there, and that one's called E, right? So just talking about Windows, so data acquisition is almost always done under Windows. You have to do it under Windows, and the real reason why is because Windows has the best support for device drivers, and what'll always happen if you try to do your acquisition on Mac or Linux is that then eventually you need to add a camera or something, and the camera only has Windows drivers, or some sort of sound thing, and then it only has Windows drivers. So you basically we're stuck using Windows. So so you have these drive letters, but the drive letters can correspond to different physical drives, or they could correspond to different partitions within the same drive, right? So one of the things is that that people can do that's a bad idea. So let me put it this way. So so you have what's called your boot partition. And that's the partition you boot off of that actually contains all the Windows stuff, right? And the Windows system file stuff. If that partition fills up, your system will crash immediately, and it might actually even be difficult to recover it. So what you need to do if you're going to do experiments and you're going to record data, what I actually recommend that you just have, so that you have your boot partition and stuff, and then that'll be just the boot drive, and actually this is typically an SSD because you'll get faster performance if you run so if you have your boot stuff and all your programs on here on an SSD, and then you can put in spinning disk hard drives in your system, and each of those will be just for data storage, right? So when you run an experiment, you save to this drive. What happens is that when you save to that drive goes and gets full. If this drive fills up, then your software might crash and tell you that everything's full, but your computer isn't going to crash. And another thing about this, which is relevant in terms of thinking about partitions, is that hard disks are so hard disk spinning disk hard drives actually have they actually have like a spinning platter with a head that like moves around. So if you try to access one file from a hard disk, then you'll access it at a certain rate. But if you want to access two files from a hard disk at the same time, so say one of the files is like stored there and the other one's stored there, the thing actually has to physically go back and forth between those two things. So copying two files off a hard disk is much more than twice as slow as copying one file off a hard disk or writing one file. So so if you have multiple partitions, so say that you had this split up into partitions E and F, and they're on the same physical drive, and then you start copying files from E and F, or start accessing E while you're using F or something like that, then this thing's going to grind to a halt. I mean, not really, but it's going to get really slow. So so what I recommend is best if you have everything. So if you are going, so in my lab we actually have these small computers, so we do everything on SSDs and there's no spinning disks involved. So in those computers, what you really should do is configure it so that you have a separate boot partition that contains all the programs and probably your user, like your desktop and stuff, and then have it so you have a second partition that's just for data, and then you save your data into that partition. So even if it fills up, you don't crash your entire system. And it's like you can crash your entire system and it won't boot again. You know what I mean? So it's something where you actually have to boot off some sort of rescue disk and clean it up, and it's a mess, right? So so try to have separate partitions you save stuff in. Something like this, where you have a hard disk that's split up into multiple partitions. One of the things that's happened to me before was that if two people are using the same rig, and one person is recording, and then the other person wants to copy their data off over the network, and if both of those are, say, different partitions are on the same hard disk, then so this happened to me because I was copying data off someone like with Lisa Rue, who I was sharing a rig with as a postdoc. And I copied data when she was recording, and it was on the same drive, and her experiment crashed. And she got really pissed at me. So what's actually much better is that if each individual person, so I mean, there's many different ways to do it. I'll tell you the way that I do it, which is that you have a system that has multiple slots for hard drives, and this is Claire's hard drive, right? And this is Luke's hard drive, right? And Claire saves to that one, I save to this one. And if I copy my data off of that one while she's recording on this one, so since the rate limiting step is the speed of the access to the physical spinning platter hard drives, like the computers and the buses and network, all that stuff is much faster, then that's going to be totally fine, unless you're doing some sort of crazy high-speed imaging camera stuff, right? But when you try to access a hard disk, more than one person simultaneously, then it just gets super, super slow, right? And then what I typically do is have these drives, and then when you fill one up, you pull it out and put it on a shelf somewhere, and then that could be also sort of like another backup, and then you put it in a new drive and you record till that one's full. Okay. I think we are out of time. I didn't think this was actually going to go 2 hours, but it actually did. Any final questions? Right, so hard disks, 100% failure rate. Hard disks are slow. You have to save your data locally, not over the network when you're doing an experiment, and then you have to copy it over the network later. Try to do everything off SSDs in terms of your actual processing and data analysis, because it's much faster. And that's pretty much it. Boot partitions, keep your boot partition separate from data partition. All right. Okay, so next Tuesday, we're going to be talking about electrophysiology. So all these general principles we discussed about filtering and sampling, electrophysiology is just that with an electrode. So as you'll see, okay? All right. Bye, everyone. See you next week.
Bye.
Thanks, bye.
Here.